Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Pondicherry University MSc Entrance Exam question paper of Biotechnology subject. It was conducted in the year 2012. So coming to the first question, an aromatic amino acid. So out of these options, which is the aromatic amino acid? Lysine, tyrosine, taurine, arginine. Lysine and arginine are not aromatic amino acids. Only tyrosine is aromatic amino acid. So answer is B. A true statement about solutions of amino acids at physiological pH is. Answer is all amino acids contain both positive and negative charges. So at physiological pH they contain both positive and negative charges. Then sulfur containing amino acid is methionine. So answer is option A. Another is cysteine. So sulfur containing amino acids, one is methionine and the other is cysteine. Then restriction endonucleases which recognize and cut same recognition sequence are known as. So they are known as isoschisomers, whereas isozymes are enzymes that differ in amino acid sequence but catalyze the same chemical reaction. So here it is restriction endonucleases which cut the same recognition sequence. They are isoschisomers, whereas isozymes are enzymes that differ in amino acid sequence but catalyze the same chemical reaction. And what are abzymes? Abzymes are antibodies that catalyze specific chemical reaction, function as enzymes. Abzymes are also called as CatMab or catalytic monoclonal antibodies. Coming to the next question, a radioactive element is sometimes used to trace the pathway of chemical reactions the cell. If newly synthesized proteins are radioactive, the radioactive element could be. Answer is option C, nitrogen. Sodium is not present in any of the amino acids, neither is chlorine nor potassium, but nitrogen is present in amino acids. So the newly synthesized protein are radioactive. The radioactive element could be on the nitrogen because proteins are made up of amino acids. So nitrogen is present in amino acids and the radioactive element will be incorporated in the nitrogen. Then next one, a net gain of water tend to occur in a hyperosmotic solution from an hypoosmotic solution. So when you play, a net gain of water occurs in a hyperosmotic solution from a hypoosmotic solution. Water will enter into the hyperosmotic solution from the hypoosmotic solution. Then many surface proteins are anchored by that is on the cell membranes. They are anchored by non-polar amino acids because the lipid bilayer, the lipid fatty acid chains are non-polar. So the proteins must anchor into the non-polar bilayer. So they should have hydrophobic or non-polar amino acids in their sequence. So they are anchored to the lipid bilayer by non-polar amino acids. A, B, O blood groups are marked by surface. They have surface glycoproteins. So answer is option A. Then next one. The number of subclass of IgG is. IgG has 4 subclasses. They are IgG1, IgG2, IgG3 and IgG4. Now PTHs reduce the renal clearance or excretion of calcium, increases renal phosphate clearance, increases renal clearance of calcium, decreases the renal phosphate clearance. So now PTH stands for parathyroid hormone. It is a protein hormone released by the parathyroid gland. It raises calcium levels by releasing calcium from bones and increasing the amount of calcium absorbed from your small intestine. Now what is renal clearance? Renal clearance of a substance refers to how quickly a particular substance is removed from the plasma by the kidney and excreted in the urine. So what is renal clearance? It refers to how quickly a particular substance, maybe calcium or glucose or any other ions, is removed from the plasma by the kidney and excreted in the urine. So PTH is, as per this option, it increases renal phosphate clearance because parathyroid hormone decreases phosphate reabsorption. So it increases renal phosphate clearance. Then next one, delta cells of islets of Langerhans of pancreas produce. They produce the hormone somatostatin. Answer is C. A microscope in which an image is formed by passing an electron beam through a specimen and focusing the scattered electrons with magnetic lens is called. This is the carried out by using the scanning electron microscope. So answer is B. Then next one, transmission electron microscopy is best for high magnification viewing of. They are used for internal structure of fixed cells. Answer is option A. Then next one, to make a vaccine against chicken cholera that would not kill the chicken, Pasteur. So what did Pasteur do? Treated the sample with heat to kill the microorganism, attenuated the strain by repeatedly passaging it in culture, used a related but different microorganism from animals, used very small non-lethal amounts of material. So to make a vaccine against chicken cholera that would not kill the chicken, what did Pasteur do? He attenuated the strain by repeatedly passaging it in culture. So answer is option B. The first observation that bacteria-like organism could be found in normal air was by Louis Pasteur. So answer is option B. Then, which of the following is not equivalent to 10 micrometers? So they have given centimeter, nanometer, millimeter and angstrom units, and they have asked which is not equivalent to 10 micrometers. So 10 micrometer will be 10 into 10 raised to minus 6 meter, and 1 meter is 100 centimeter. So in option A, they have given the unit as centimeter. So we are converting it into centimeter. So 10 into 10 raised to minus 6 meters will be 0.001 centimeter. So the answer is A, 0.001 centimeter. But we will look at the other options also. Then option B, the unit is millimeter. So convert it into millimeters. So 1 meter will be equal to 1000 millimeters. So 10 into 10 raised to minus 6 meters will be equal to 0.01 millimeter. So B is correct. Now coming to the next option, that is C, which is nanometer unit. So 1 meter is equal to 10 raised to 9 nanometers. So 10 into 10 raised to minus 6 meters will be equal to 10,000 nanometers. So option C is also correct. Then coming to option D, the unit is angstrom units. So 1 meter is equal to 10 raised to 10 angstrom units. So 10 into 10 raised to minus 6 meters will be 100,000 angstrom. So the one which is not equivalent to 10 micrometers is 0.001 centimeter. It is equivalent to 0.001 centimeter. So answer is option A. Then next one, bacteria accomplish chemotaxis by. So the answer is frequently stopping and tumbling to better sense good conditions. So that is how bacteria accomplish chemotaxis. When two ecosystems overlap each other, the area is called an ecotone. Ecotone is a transition area between two biomes. It is where two ecosystems overlap each other. It may be narrow or wide, and it may be local also or regional also. If it is local, example will be the zone between forest and a field. And if it is regional, the transition between forest and grassland ecosystems. So answer is, where the two ecosystems overlap, it will be ecotone. Then next one. Now, okay, before that we will see what is habitat. Habitat is the natural home or environment of an animal, plant or other organism, or place where the organism make its home is called a habitat. Whereas niche in ecology is all of the interaction of a species with the other members of its community, including for competition, predation, parasitism and mutualism. An ecological niche refers to the interaction of a species with all the biotic and abiotic factors affecting it. And what is an ecotype? A distinct form or race of a plant or animal species occupying a particular habitat is called as an ecotype. Then next one. Which of the following two hormones are essential for induced breeding of fishes? Now, what is induced breeding? It is a technique by which economically important fish are bred through artificial stimulation of pituitary injection. So, the hormones essential for induced breeding are FSH and LH. Answer is option C. Then ACTH induces rise in. It will induce a rise in cyclic AMP levels. So, answer is option A. Then, the transgenic animals are for improvement of the quality of milk, meat, eggs. All of the above is the answer. Transgenic animals can be used for improvement of the quality of milk. Then it can be used for meat improvement and also for eggs. That is to improve animal products for human consumption. So, for all these we can use transgenic animals. Then hormones. Hormones act as coenzymes. That is correct. So, answer is option A. The movement of sodium ions from an area of higher concentration to an area of lower concentration is called diffusion. Answer is option C. Whereas active transport, it is a kind of transport where substance moves against a concentration gradient. That is from an area of lower concentration to an higher concentration. This process also requires expenditure of energy. Then osmosis is the process by which molecules of a solvent tend to pass through a semi-permeable membrane from a lesser concentration solution to a more concentrated one. So answer is option C, that is diffusion. Then during cytokinesis in plants. During cytokinesis, a bundle of actin microfilaments called the contractile ring pinch the cell in half. No. Then they have asked it in plants. Small vesicles directed by phragmoplast move to the spindle. That is correct. So during cytokinesis in plants, small vesicles directed by the phragmoplast move to the spindle. Then next one. During the RAS pathway, cytoplasmic protein kinases are activated. The growth factor receptor is dephosphorylated. No, it is phosphorylated. Then growth factors bind to receptors in the cytoplasm. Not in the cytoplasm but on the plasma membrane. Leads to the production of translation factors. Not production but activation of translation factors or transcription factors. So answer is A, cytoplasmic protein kinases are activated. Sister chromatids. A sister chromatid refers to the identical copies, that is chromatids, formed by the DNA replication of a chromosome with both copies joined together by a common centromere. So sister chromatids are created when DNA is replicated, yes. Are separated during mitosis, yes. Are attached at the centromere prior to division, yes. So answer is all of the above are correct with regards to sister chromatids. Then next one, DNA damaged by sunlight. The DNA damage will sunlight will have pyrimidine dimers. So answer is option C, has formed pyrimidine dimers. Then IgA antibody is the first line of defense against infections at the mucous membrane. It is usually an early specific antibody. Which of the following statements regarding IgA is not true? Complement fixation test for IgA antibody will be positive if specific IgA antibody is present. That is correct. IgA is not found in saliva, therefore an IgA diagnostic test on saliva would have no value. IgA is present in saliva, tears, and therefore an IgA diagnostic test on saliva would have some value. Then IgA can be destroyed by bacterial proteases. So there are three major bacteria, that is Neisseria gonorrhea, Streptococcus pneumonia and Haemophilus influenza type B. They release IgA protease which destroys IgA. So that is also correct. IgA is absent in colostrum. So that's also correct. So answer is option B. Then next one. An immunoglobulin molecule always contains 1 kappa and 3 lambda chains, 2 kappa and 2 lambda chains, 3 kappa and 1 lambda type of chain, 2 kappa and 2 lambda chains. So it contains 1 kappa and 3 lambda type of chain. Then next one. A flower which can be divided into two equal halves by only one plane. So what is called a zygomorphic flower. Now what is actinomorphic? Actinomorphic is when a flower can be partitioned into two equal radial halves in any radial plane passing by the center. So it can be divided into two equal halves along any radius. Then a regular flower, when all the parts of each whole of a flower, that is petals, are in similar shape and sizes, then such a flower is called regular flower. And what is a perfect flower? Is one in which both male and female reproductive structures are present. It's called a perfect flower. Then next one. Which of the following bacterium is used for production of transgenic plants? So Agrobacterium tumefaciens, that is used for the production of transgenic plants. Then spindle fiber is made of, they are made of tubulin protein. So answer is option D. Then co-repressors, what does they do? They trigger the shutdown of gene transcription, not translation. So answer is option B. They are co-repressors. The theory that organelles have evolved from bacteria engulfed by a long ago cell is called as endosymbiont theory. Answer is option C. They have told that as per the endosymbiont theory, chloroplast and mitochondria have evolved from bacteria engulfed by a long ago cell. So that is called as endosymbiont theory. Then both mitochondrial DNA and chloroplast DNA are overwhelmingly inherited. They are inherited from the maternal side. That is, your ovum or the egg will contain the mitochondrial DNA. So answer is A, from the maternal side. Who first described microorganisms such as bacteria? It was described by Anton von Leeuwenhoek. Credit for the first vaccine for the prevention of human disease is generally given to. It is given to Edward Jenner for the prevention of smallpox. Then hyperglycemic effect of glucocorticoids is due to. Glucocorticoids oppose insulin action and stimulate gluconeogenesis. So stimulation of synthesis of uh, L-troxykinase. Stimulation of, so that is the answer, stimulation of synthesis of L-troxykinase. So hyperglycemic effect of glucocorticoids is due to stimulation of synthesis of L-troxykinase. Then, which immunoglobulin in the primary antibody in saliva, tears and intestinal genital secretions? So it is IgA that is present in saliva, tears and intestinal and genital secretions. Then what is ribosine? Not a regular enzyme but has the action of an enzyme. That's correct. Catalyzes cell splicing of pre-tRNA segment. That is also correct. Self-splicing of RNA is also found in rRNA, mRNA, chloroplast tRNA, rRNA and mRNA. So ribozymes are found in, ribozymes are all these. So all of the above is the answer. The next one. According to fluid mosaic model, the correct sequence of plasma membrane is protein, then you have the lipid bilayer and again protein. So answer is option C, protein, lipid, lipid, protein. The number of chromosomes can be increased in plants by applying. They can be increased by colchicine treatment that will inhibit spindle fiber formations. So answer is option C. Then unique property of DNA polymerase 1 is it has got 5' to 3' exonuclease activity and it is involved in repairing of during replication of DNA during replication. So it has got 5', 3' exonuclease activity. Then next one. An important difference between eukaryotic and prokaryotic replication is eukaryotic DNA polymerases are faster. More DNA polymerases are found in eukaryotes. Multiple origin of replication in eukaryotes. Answer is eukaryotes have multiple origin of replication when compared to prokaryotes because their chromosomes are more longer than that of the prokaryotes. So in order to synthesize a new one, they should have multiple origins. Only then they will be able to finish it in less time. So answer is multiple origin of replication in eukaryotes. Then which system for stably introducing foreign genes into eukaryotic cells does not involve the integration of the foreign DNA into the host chromosome? Answer is option B, transgenic mice. Whereas in all the other system, stably introduction of foreign DNA into eukaryotic cells take place or integration of the foreign DNA into the host chromosome take place, but not in case of transgenic mice. Then approximately how many moles of ATP will be generated as a result of oxidation of 1 mole of FADH2? Answer is option A, 2. But now the current values are 2.5 ATP per NADH and 1.5 ATP per FADH2. Then which of the following is not encoded by MHC genes? MHC genes don't encode immunoglobulins. Answer is option B. The next one, which is least likely to occur for removal of cancer cells? T cell based cytotoxicity, that will be there in case of cancer cells, complement fixation and phagocytosis, but autophagy doesn't result. Then regulation of tryptophan operon by binding of tryptophan to tryptophan receptor is termed as repression. Answer is option A. Oxygenase activity of Rubisco generates. It generates 1 molecule each of PGA and phosphoglycolate. So answer is option C. Oxygenase activity means when Rubisco binds oxygen. Rubisco has got affinity towards carbon dioxide as well as oxygen. So during the oxygenase activity, it will generate one molecule each of PGA and phosphoglycolate. Then which is true for beta oxidation of fatty acids? Formation of malonyl CoA, formation of acetoacetyl ACP. So formation of malonyl CoA is for fatty acid synthesis. Formation of acetoacetyl ACP is also fatty acid synthesis. Then transport of acyl CoA into mitochondria. That is correct for beta oxidation of fatty acids. Use of NADPH2. It uses NAD, not NADPH2. Then enzymes do not interfere with. Enzymes do not interfere with free energy of the reaction. That is correct. Rate of the reaction. By increasing the enzyme concentration, the maximum reaction rate also increases. Then enzymes can greatly speed up the rate of reaction by reducing the activation energy needed to make the reaction get started. Then enzymes also do not alter the equilibrium point of the reaction, or they do not change the equilibrium constant for a reaction, or it can be told that they do not affect the Gibbs free energy of a reaction. So enzymes do not interfere with reaction equilibrium as well as free energy of a reaction. So both A and D are correct. Then next one, I don't know what was given as option in your answer key. Then next one, covalent bond formation between two atoms take place, electrons sharing by both interacting atoms. So answer is option C. Then the main difference between cellulose and starch molecule, that is the type of linkage between glucose subunits. In cellulose, in an unbranched polymer of cellulose is an unbranched polymer of glucose residues joined by beta-1,4 linkages, whereas starch is found in two forms, that is amylose, that is unbranched type of starch and has alpha-1,4 linkages for glucose residues. And another form of starch is amylopectin, that is the branched form and has about alpha-1,6 linkages per 30 alpha-1,4 linkages. That is, for every 30 alpha-1,6 linkages there is a branch and it has got alpha-1,4 linkages. So it is on the type of linkage between glucose units that cellulose and starch molecules differ. Then two functions of rough endoplasmic reticulum are to detoxify and transport drugs, modify and activate hormones, synthesize and transport enzymes. So answer is option C. Then next one, all of the following are correct about the X and Y chromosome except there are 78 active genes on the Y chromosome. Females have one X and one Y chromosome. No, females have both X chromosomes only. There is no Y chromosome. In early meiosis, identical chromosomes can repair mutation. X and Y chromosomes cannot pair and exchange genetic information. So all the others are correct except females have one X and one Y chromosome. In many countries, DDT is banned as an insecticide because it is not biodegradable. So answer is option B. Then, in the process of Krebs cycle, ADP is converted into ATP, pyruvic acid is converted into ATP, acetyl-CoA is converted into carbon dioxide and water, glucose is converted into carbon dioxide. So, in Krebs cycle, ADP is converted into ATP. So, answer is option A. Then, with regards to HLA class 1 antigen, which of the following is below is false? They are expressed on all nucleated cells. That's correct. They are made up of heavy chain and a light chain. Yes. They are essential for viral antigen recognition by cytotoxic cells. That is also correct. The genes for HLA class 1 molecule are located on chromosome 6 and 15. They are located on chromosome 6 but not on 15. So answer is option D. Then next one. In the hybridoma technology, the marker enzyme responsible for selection of hybridoma cells, that is HGPRT. Answer is option B. Hybridoma is for the production of monoclonal antibodies. Then erucic acid is present. It is present in mustard oil. Answer is option A. Expansion of BLAST. BLAST is Basic Local Alignment Search Tool. So there is some printing mistake in the given options. B will be more correct. It must be Basic Local Alignment Search Tool, not logical. Then next one. Example for DNA virus. Cholineviruses are DNA viruses. So answer is option A. Then reverse transcriptase catalyzes the formation of DNA on an RNA template. So answer is option B. Then bacteriospores. They are resistant to antibiotics. So that's correct. Allow the bacteria to multiply in adverse condition. So it allows bacterium to produce a dormant and highly resistant cell to preserve the cell's genetic materials in times of extreme stress. So they don't let the bacteria multiply, but they try to ward off the extreme stress in the environment. Then are usually formed by gram-negative bacteria. Only gram-positive bacteria can form spores. So 3 is also not correct. Can be identified with gram stains, are killed by temperature at 120 degrees Celsius for 10 minutes. So 1 and 5 are only true. So answer is option A. zDNA is characterized by, zDNA is a left-handed helix. So answer is option A. Nickel and chromium compounds can cause cancer of the lungs. Answer is option B. Then, which of the following is not formed during anaerobic respiration? During anaerobic respiration, pyruvic acid is there, ethyl alcohol is formed, carbon dioxide is released, but acetyl CoA is not produced. So, answer is option D. Type of chemical bond between glucose and fructose in sucrose. It is alpha-1,2 glycosidic bond. So answer is option C. Then approximately what percentage of DNA in the human genome is both transcribed and translated? It's only 3%. So answer is option D. In a charged transfer RNA, the nucleotide bound to the amino acid is adenosine and the next two nucleotides are cytosine. What can you tell about the DNA codon to which this transfer RNA corresponds? Now, the first position is A, but you can't tell about the others from the information given. So, it is a charged tRNA. The nucleotide bound to the amino acid is adenosine. So, nucleotide bound to the amino acid is adenosine and the next two nucleotides are cytosine. So, in a charged RNA, the nucleotide is bound to the CAA. I think if I am correct, or I think it is CAA is correct, not AAC. Okay, CAA is correct. So, the amino acid is bound over there. Bound to the adenosine and the next two nucleotides are cytosine. So CAA is correct. So in the charged tRNA, it is bound to that bound to that arm of the tRNA. Then what can you tell about the DNA codon to which transfer RNA corresponds? They have not told which amino acid is bound to this adenine. So we cannot tell anything about the DNA codon to which this transfer RNA corresponds. So answer is option B. You can't tell anything about the codon from the information given, because it is only told that in charged tRNA, the nucleotide bound to the amino acid is adenosine. So nucleotide of the tRNA, that is adenosine, is bound to the amino acid. But they have not told which amino acid, whether it is alanine, lysine, glycine, or out of those 20, which amino acid is bound, they have not told. So we cannot predict what will be the DNA codon to which this transfer RNA corresponds. So answer is option B. Then, what is the main target of natural selection? Answer is option A, that is the population. The next one. Below are listed a few cell organelles. Organelles not enclosed by two phospholipid membranes are. So those which have only a single phospholipid membrane, not double membrane. So nucleus is having double, then lysosomes, it is single, endoplasmic reticulum also single, peroxisomes single, mitochondria double, and chloroplast double. So the answer is 2, 3 and 4. Answer is option C. Then hexose monophosphate shunt is an additional pathway for oxidation of glucose. It generates NADPH, essential for fatty acid synthesis. This pathway predominates in all except one of the following tissue. The tissue it is found in liver, adipose tissue, adrenal cortex, and mammary glands. But it is not found in skeletal muscles. So answer is option D. And why does skeletal muscle have low expression? Because the HMP because of the presence of low levels of expression of enzymes of HMP pathway. Then the lowest level of activity of superoxide dismutase and catalase is found in obligate anaerobes. Obligate anaerobes lack superoxide dismutase and catalase and or peroxidase, and therefore undergo lethal oxidation by various oxygen radicals when they are exposed to oxygen. So it is found, the lowest level is found in case of obligate anaerobes. Then gibberellins are known as to break dormancy in cereal seeds. This dominantly is due to secretion of alpha amylase. Answer is option C. Then bacterial rope protein is a helicase. Answer is option A. Zinc fingers are important in cellular recognition because they are structural motifs in many DNA binding proteins. Answer is option C. So next one, the enzyme that changes ribulose to cellulose is it is an epimerase. So answer is option C, epimerase. L-ribulose 5-phosphate 4-epimerase is an enzyme that catalyzes the interconversion of ribulose 5-phosphate and cellulose 5-phosphate in the oxidative phase of pentose phosphate pathway. Vitamin A is stored in the liver in the form of retinyl palmitate. Answer is option D. A mutation deleting an upstream activating sequence for a single gene would be expected to be cis dominant. The next one, in vertebrate genes, transcription regulatory regions that contain CpG islands are inactivated by the CpG modification. So what are CpG islands? They are short interspersed DNA sequences that deviate significantly from the average genomic pattern by being GC rich. So these CpG islands are GC rich, G or CpG rich, and predominantly non-methylated. So cytosines in this CpG dinucleotides can be methylated to form 5-methyl cytosines. So answer is option D, that is methylation. Then next one, 2-amino 6-oxopurine is, that is 2-amino 6-oxopurine is guanine. Answer is option C. The rate limiting step of fatty acid synthesis is catalyzed by. The rate limiting step in fatty acid synthesis is the formation of malonyl CoA from acetyl CoA by the enzyme acetyl CoA carboxylase. So answer is option A. Next one. In a hybridization experiment, a plant shows phenotypic ratio of 15:1. How many genes control the trait for observed phenotypic ratio? Now, complete dominance at both gene pairs. However, when either gene is dominant, it hides the effect of the other gene. So, duplicate dominant epistasis. So, 15:1 is a ratio of duplicate dominant epistasis. So, answer is option B, that is 2. 2 genes control the trait for the observed phenotypic ratio. Parkinson's disease is associated with. It is associated with an underproduction of dopamine. It is a progressive disorder that causes the degeneration of nerve cells in the part of the brain called the substantia nigra, which controls movement. These nerve cells die or become impaired, losing the ability to produce an important chemical called as dopamine. So answer is option B, an underproduction of dopamine. Now, a restriction endonuclease that recognizes the base pair sequence GANTC, where N can be any nucleotide, will cleave random DNA on average. It will cleave on an average 4 raised to 5. So that will be 1024 base pairs. So answer is option C. Then, which of the following is not a cytokine? Arachidonic acid. Arachidonic acid is not a cytokine, it is a fatty acid. Interleukins are cytokines, interferons are cytokines, and tumor necrosis factor is also a cytokine. Next one. Identify the following point mutation in. So this is a repeated question, and I have already discussed this in the previous question paper. You may refer to the previous question paper for its explanation. The answer is frame shift and transition respectively. So answer is option B. Which of the following is not a trait of an anabolism in the metabolism process? The process of breaking down complex molecules to release energy, that is not a trait of anabolism. This is also a repeated question. A shiny sticky colony of Streptococcus pneumoniae is likely to be. It is likely to be encapsulated and pathogenic. Answer is option C. Then reversal UV effect is called as. It is called as photoreactivation. Answer is option D. Then liposomes are used in. Liposome is a tiny bubble or a vesicle made out of the same material as a cell membrane. Liposome can be filled with drugs and used to deliver drugs for cancer and other diseases. So liposomes can be used in drug delivery. Answer is option A. NSAIDs are used as. NSAIDs are non-steroidal anti-inflammatory drugs. That is the full form of NSAIDs. And these are medicines that are widely used to relieve pain, reduce inflammation, and bring down a high temperature. So answer is anti-inflammatory agents. Option B. Then CCA end of the tRNA molecule is called as. It is called as the acceptor arm. It is where the amino acid binds. The amino acid binds to the adenosine of the tRNA molecule. So it is the acceptor arm. Then Tata box is present at, it is present at minus 30. Answer is option A. The catabolic end product of purines in humans is, it is, answer is option B, uric acid. Then next one, the percentage of triglycerides is more in, it is more in kilomicrons. So answer is option C. Then, which of the following is not part of the lac operon of E. coli? Genes for inducible enzymes of lactose metabolism, that is there. Then genes for the repressor regulatory protein, that is also part of lac operon. Gene for RNA polymerase, that is not present in lac operon. So answer is option C. Then next option, a promoter, the RNA polymerase binding site. So that's also present in lac operon. What is the difference between apoptosis and necrosis? Apoptosis is controlled program of cellular destruction and necrosis is cell death due to damage. So answer is option A itself. So thank you for watching, and please like, share and subscribe. Thank you.